14. Properties of the cardiac muscle. Principle. The properties of the cardiac muscle are studied in a beating heart and a quiescent heart. We start by learning about extrasystole and the compensatory pause. We record the normal cardiogram on the kymograph. Then we apply the stimulus during different phases of the cardiac cycle and observe the graph obtained. When we apply a stimulus to the heart in systole, there is no reaction to the stimulus. The cardiogram remains unchanged. When we apply the stimulus during diastole, we can see that there is an extra curve in the graph followed by a pause. This is because the stimulus generates a premature contraction or systole in the ventricles. This is called an extrasystole. The extrasystole is followed by a compensatory pause. In the graph, we can see a small section where there are no curves immediately after the extrasystole. This is called the compensatory pause. The compensatory pause is seen because the next normal impulse from the sinus venosus falls in the refractory period of the extrasystole. The contraction after the extrasystole is stronger because calcium ions accumulate in the muscle fibers during the compensatory pause. Let us now learn about the refractory period. We can understand the refractory period better on a quiescent heart. We make the frog's heart quiescent by tying a ligature tightly between the sinus venosus and the atria. This prevents the impulses from the sinus venosus from reaching the atria or ventricles, and for some time the atria and ventricle do not contract. After we make the heart quiescent, we apply electrodes to stimulate the ventricle. In the first scenario, we apply the first stimulus and let the ventricles contract, and then when the ventricles are completely relaxed, we apply the second stimulus. We record the resultant graph. Then we reduce the angle between the two contact arms so that the second stimulus is applied while the ventricles are relaxing. We record the graph. Finally, we further reduce the angle such that the second stimulus is applied while the ventricles are still contracting. We record the graph. In the first graph, when the second stimulus is applied after complete relaxation, the ventricles respond to the second stimulus and contract again. In the second graph, when the second stimulus is applied while the ventricles are relaxing, a strong second stimulus can generate a response. This is because the second stimulus falls in the relative refractory period of the heart muscle. The relative refractory period of the heart muscle lasts for 50 milliseconds. In the relative refractory period, while the muscle is relaxing, a strong stimulus can elicit a response. In the third graph, we apply the second stimulus during the period of contraction of the ventricle. This is called the absolute refractory period. During this period, no stimulus, however strong, can elicit a response from the cardiac muscle. Cardiac muscles have a very long absolute refractory period. It lasts for almost 250 milliseconds. Cardiac muscles have a long refractory period because the action potential of the cardiac muscle lasts for almost as long as its mechanical contraction. Here is a graph where you can see the duration of the action potential and the duration of the muscle contraction. We see that the duration of action potential is almost equal to the duration of the muscle contraction. Because of this long duration of action potential, cardiac muscles cannot be tetanized. Now, let's try and understand the all or none phenomenon. To understand this, we arrange the experiment like the experiment to record the normal cardiogram. After tying the first Stanius ligature to make the heart quiescent, we apply electrodes to the base of the ventricle. We stimulate the ventricle with a subthreshold stimulus and observe the graph. We gradually increase the strength of the stimulus with each consecutive stimulus till a contraction is seen. We keep an interval of 30 seconds between every two stimuli. We keep increasing the strength of stimulus even after the first contraction to observe for any change. We study the resultant graph. We can see that before the stimulus reaches threshold level, there is no contraction of the ventricle. Only after the strength of stimulus crosses the threshold level, the heart muscles start contracting. Even if we increase the strength of stimulus further after crossing the threshold, the strength of contraction does not increase. This is because the heart muscles and the heart as a whole follow the all or none law. This means that only a threshold or supra-threshold level stimulus can elicit a response from the heart muscles. And increasing the strength of stimulus above the threshold level does not correspondingly increase the strength of stimulus. Think of the staircase phenomenon. To study the staircase phenomenon, we apply stimuli rapidly to the ventricles. We make the heart quiescent by tying the first Stanius ligature and set up the apparatus. We stimulate the ventricle repeatedly at intervals of 2 seconds and record the resultant contractions on a graph on a stationary drum. We can observe that the first few contractions successively increase in amplitude before forming a plateau. This is because of a phenomenon we call the staircase phenomenon. When we stimulate the ventricular muscles repeatedly with the interval between two successive stimuli, 10 seconds or less, the contractions increase in amplitude. Summation of subminimal stimuli. We make the heart quiescent by tying the first staneous ligature. We adjust the strength of stimulus to a point that it just fails to elicit a response. Now we repeatedly stimulate the ventricle till the subthreshold stimulus elicits a response. The subminimal stimuli, when applied rapidly at an interval of less than 1 second, summate and produce a response. This is also because of the excess calcium ions remaining in the muscle